Hi Achievers, welcome to my channel. Here you will get detailed line by line explanation of NCERT biology with added wonderful images and important notes for Neet, AIIMS, JIPMER and other competitive exams. In this video, I am going to explain about the tools of biotechnology. This is part 2 video of chapter 11. Part 1 video link is in description box. Please watch it. Without any delay, let us go to the chapter. For better understanding, keep the NCERT textbook aside and also take the screenshots so that they'll help in your revision. Tools of recombinant DNA technology. As we discussed in the previous video, the recombinant DNA technology contains 3 main steps. The first one is identification of desired DNA, and the second one is introduction of this DNA inside the host, and the third one is maintenance of this desired DNA inside the host and its division into the progeny to get multiple copies. And for this to happen, the tools are very important. There are 5 tools which help in recombinant DNA technology. Coming to the first tool, these are restriction endonucleases. Restriction endonucleases help in cutting of the DNA. After cut by the restriction endonucleases, they get joined. Polymerase. Polymerase helps in polymerase chain reaction, which is an important step in the cloning of DNA molecule. Ligases. Ligases are the enzymes which join the 2 foreign DNA after they were cut by restriction endonuclease. Vectors. Vectors are the agents which transfer the desired gene into the host organism. Prominently, PBR332 is widely used plasmid vector. Host organisms. These are the organisms inside which our plasmid or recombinant DNA get multiplicated into its clones along with the desired DNA. The first tool is restriction enzymes. The isolation of restriction enzymes is a milestone in biotechnology, that too in genetic engineering. In the year 1963, the 2 scientists, Stavorlin and Werner Arber, isolated 2 enzymes. These 2 enzymes are responsible for restricting, that means stopping, the growth of bacteriophage inside the bacteria. Why these 2 enzymes are stopping the growth of bacteriophage in bacteria? Because the bacteriophage means it is a virus, enters inside the bacteria and kills the bacteria. In order to save the bacteria, it is synthesizing 2 enzymes. These 2 enzymes prevent this bacteria from bacteriophage. And how they are preventing? One of the enzyme, methylase, adding methyl groups to the bacterial DNA. And the other enzyme, restriction endonuclease, cuts the viral DNA. This is the bacteriophage or virus attacking bacteria and killing it. Here I have to cite one example. Coronavirus attacked so many people and the scientists have discovered vaccines in order to kill the coronavirus. But to prevent itself from the vaccines, the coronavirus got mutated and resulted in different strains. In the same way, after several years, in order to prevent the attack by these viruses, the bacteria synthesize 2 enzymes. The first one is methylase, which adds methyl groups, and the second one is restriction enzymes. These restriction enzymes will get activated after the virus attack, and then it cuts the DNA of the virus and kills it. Thus, it protects its own bacteria from the bacteriophage. And here, as the restriction enzyme killing DNA of the virus, why can't it kill its own bacterial DNA? It also can be possible, right? So, in order to prevent it, the methylase adds methyl groups to the bacterial DNA so that the restriction enzyme can't digest or cut its own bacterial DNA. For suppose, this is the bacterial DNA, and when bacteriophage attacks the bacteria, First, the methylase got activated and it adds methyl groups to the bacterial DNA. Then restriction enzyme gets activated and it comes to the bacterial DNA. It identifies its restriction recognition site, but here as there are methyl groups, it can't recognize its restriction site and it doesn't cut the bacterial DNA. And it goes to the viral DNA. There are no methyl groups. So it recognizes its recognition site and cuts the viral DNA, killing it. This is the process of protecting bacteria from the bacteriophage attack by the two enzymes methylase and restriction endonuclease. From then, various restriction enzymes were isolated, and the first restriction endonuclease used in recombinant DNA technology is HIND2, whose functioning depended on a specific DNA nucleotide sequence, is isolated and characterized 5 years later. HIND2 acted on the double-stranded DNA where there is a specific sequence. See, this is a double-stranded DNA, and in these sites from 5' to 3' direction, the GTAC, 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 and in all these sites the sequence is same, GTAC. Here also in 3' to 5' direction, CATG, CATG, and HIND2 recognizes this sequence and cuts at the middle of this sequence. See, this is site 1, site 2, site 3, site 4 and so on. In this way, after recognizing a particular sequence, it cuts. If it cuts at the middle of the sequence, blunt ends will be formed. And if it cuts away from the middle of the sequence, the sticky ends will be formed. That we will discuss later. Apart from HIND2, scientists have isolated over 900 restriction enzymes from over 230 strains. More than one restriction enzyme can be isolated from a single bacterium. All these 900 restriction enzymes contain specific recognition site. Each enzyme's recognition sequence is different from the other enzyme. And the convention of naming these enzymes is the first letter of the name comes from the genus. Take Echo R1 as an example. The first letter E comes from its genetic name Escherichia. And the second two letters come from the species. The second two letters CO is named according to its species name Coli. And the third letter refers to the strain or Y13. And 1 refers to the order of its isolation. That is, Echo R1 is first isolated from bacteria Escherichia coli. So it is named as Echo R1. And in the same way, HIND2, see the structure of HIND2. HIND2 is named according to this nomenclature as it was extracted from Haemophilus influenza. H refers to its genus name Haemophilus. IN, the two second and third letters, refers to the species name Influenza. D refers to its strain name and 2 refers to its order of isolation. It is isolated second. It is the second enzyme that is isolated from Haemophilus influenza. Enzymes are classified into several types, and these restriction endonucleases come under the classification of enzymes, that is nucleases. Nucleases are the enzymes that cut the nucleotide bases, and these are of two kinds, exonucleases and endonucleases. Exonucleases are the enzymes that cut the nucleotides from the ends of the DNA. See here, exonucleases are cutting from the ends of the DNA and removing the nucleotides, whereas endonucleases make cuts inside the DNA at a specific position within the DNA. These are called endonucleases. Each restriction endonuclease functions by inspecting the length of DNA sequences. First, after adding the restriction enzyme, it comes and first it identifies its recognition sequence. See here, HIND 3 recognition sequence is AAGCTT. It comes here and identifies this recognition sequence. Once it finds its recognition sequence, it will bind to the DNA. See here, once it finds its recognition sequence, it will bind to the DNA and cut each of the 2 strands of double helix. After recognizing this sequence, it will bind to the DNA and then it will cut this nucleotide sequence at a particular point. And each restriction endonuclease recognizes its sequence of palindromic nucleotides. See, what are palindromes? For suppose take the word Malayalam. The word Malayalam, when it read from left side and right side, it spells the same. And the other word is madam. If you read madam from right side, M-A-D-A-M. And from left side also, it is same, M-A-D-A-M. In the same way, it is a palindrome in words. But in DNA, the palindrome means the nucleotide bases when read from 5' to 3' direction, it is same. For example, the recognition site of echo R1 is GAATTC from 5' to 3'. And the opposite strand is from 3' to 5'. And the complementary to G is C. The complementary to A is T. Here T, A, G. And if you read from 5' to 3' in the lower strand, it is also same, GAATTC. This is known as palindrome. Palindromes are the sequences which reads the same when read from 5' to 3' direction. Observe this diagram. Here, the echo R1 restriction endonucleases, after it is added, it comes here and it recognizes its site both in vector DNA and also in the foreign DNA. After recognizing its site, it comes and cut between G and A in the both strands. Cuts between G and A in the both strands. It resulted in the formation of sticky ends. What are these sticky ends? See, here the ends are formed like this. Hanging ends are formed. These are called sticky ends. Why they are called sticky ends? Because after cut by the restriction enzyme, they again can be joined easily with the formation of hydrogen bonds, because in between A and T, 2 hydrogen bonds can be formed, and in between C and G, 3 hydrogen bonds can be formed, right? It is easy to form hydrogen bonds than phosphodiester bonds. So these 2 sticky ends can easily get joined. So they are called sticky ends. And the joining of these sticky ends is aided by ligase enzyme, resulting in the formation of recombinant DNA. Recognition enzymes cut the DNA strand slightly away from the center so that the sticky ends will be formed. The sticky ends contain overhanging stretches and these can be easily joined by the formation of hydrogen bonds. On the other side, if the restriction enzyme cuts towards the center, the blunt ends will be formed, and for joining of these blunt ends, phosphodiester bonds should be formed. So it is difficult to join them, and that is the reason the restriction enzymes that are forming sticky ends are used in our recombinant DNA technology, and the restriction enzymes that are forming blunt ends are used in cloning. Restriction enzymes cut the strand of DNA a little away from the center of the palindrome sites, but between the same 2 base pairs on the opposite strands. This leaves single-stranded portions at the ends. There are overhanging stretches called sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. The stickiness of the ends facilitates the action of enzyme DNA ligase. I have already discussed. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources or genomes. When cut by the same restriction enzyme, the resultant DNA fragment have the same kind of sticky ends, and these can be joined together using DNA ligase. Here they are joined using DNA ligase. And in this figure, the foreign DNA is attached to the vector DNA after they are cut by the restriction endonuclease enzyme. The same restriction endonuclease enzyme is used to cut both foreign and vector DNA. And then ligase joins these two DNA, resulting in the formation of recombinant DNA. Then this recombinant DNA is transferred into the bacterial cell where it clones along with the division of our desired. These are very important for MCQs in Neet, and please take a screenshot. These are different restriction enzymes which are very important for your exam, competitive exams. First enzyme is BAMH1. It is a restriction enzyme which is isolated from the organism Bacillus amyloquaciens, and its recognition site is GGATCC, cuts between G and G. Similarly, EchoR1, derived from Escherichia coli RY13. Recognition site is GAATTC, cuts between G and A in both strands. HIME3, derived from Haemophilus influenza RD. Recognition sequence is AAGCTT, cuts between A and A. TAC1, derived from Dermophilus aquaticus. Recognition site is TCGA, cuts between T and C. There is no need that recognition sequence should contain 6 base pairs. It can also contain 4 base pairs. Example is TAC1. Assume this is an insulin desired gene. Restriction enzyme cut these gene at specific sequences, resulting in the formation of different pieces of the desired gene. In order to combine these desired gene pieces with the vector, we have to separate them. For this separation, we use a technique known as gel electrophoresis. The name itself indicates that gel is used here, and electrophoresis happens in the gel. Electrophoresis means movement of electrons or particles from one negative charge to positive charge and positive charge to negative charge. The cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged, here the DNA is negatively charged molecule. Always the negatively charged molecule attracted towards the positive charge. So the DNA moves towards the anode. They can be separated by forcing them to move towards the anode under an electric field through the medium or matrix. Nowadays, the most commonly used matrix is agarose, which is a natural polymer extracted from the seaweeds. Here, this is the setup of gel electrophoresis, and here when electric field is applied, as DNA is negatively charged molecule, it moves from cathode to anode, and inside, in order to make the DNA to move, we place agarose gel. Agarose gel is extracted from the seaweeds. Agarose gel sheets are placed inside this electric field, and depending upon their size, the DNA particles moves. Smaller fragments move faster and larger fragments move slower. The DNA fragments separate according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. Here, see in this picture, we can clearly see the bands. Smaller fragments move faster and larger fragments are behind them. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide. After the movement of DNA in that agarose gel sheet, we have to see the agarose gel sheet should be visible, so that then only we can detect the DNA molecules and we can separate them. So In order to make these agarose gel sheets visible, we will add ethidium bromide. What is that? Ethidium bromide dye. It's very important for MCQs. And this ethidium bromide gives bright orange red color to the DNA bands so that they can be visible when we apply, when we see them under the UV light. Very important bit. DNA fragments are visible when we observe the agarose gel sheets added with ethidium bromide under UV light. When we observe it under UV light, we can see these bands and we can separate them by cutting the gel sheets. And the process of this cutting is known as fusion or elution, also very important bit. By this process, we can separate the DNA fragments from the agarose gel sheets, and later these fragments are purified and used in constructing recombinant DNA by joining them with cloning vectors. The cloning vectors are the second important tool in DNA technology. We'll discuss about these cloning vectors in the next video. The answer for the second bit in the previous video is insulin. Insulin gene is extracted using recombinant DNA technology. And now see some two important bits regarding this topic. Which of the statement is correct in the context of observing DNA separated by agarose gel electrophoresis? What is the question? Question is about electrophoresis. After reading the question, first you observe from which topic it is asked. It is asked from the topic of agarose gel electrophoresis. And what they asked, they have given 4 statements and they asked us to select one correct statement. Okay. DNA can be seen in the gel electrophoresis, the DNA can be seen in number 1, visible light, B, without visible light, C, when added ethidium bromide in visible light, and 4, when added ethidium bromide in UV light. I have mentioned it. We can only see DNA when we add ethidium bromide, and we can see it under UV light only. When exposed to UV light only, we can see the DNA, but not in visible light. The answer is D. Second question. Choose the wrong statement about restricting enzymes. They cut the DNA at recognition site. They cut the DNA at the center of the palindrome. They are called molecular scissors. HIND2 is the first restriction enzyme. Text the answer for this question in the comment box. Meet you soon in the next video.